Good evening, everyone. So, this is Kanika Gupta, your TA for the session, and I'll be helping you guys with your doubts from last week's lectures. So, in the last week, we studied about the translational part. If you guys remember, in the central dogma of biology, we read about three processes: the replication, transcription and translation. So we have studied already about replication and transcription. Now we are coming to the last part of the central dogma of biology, that is translation. So today we'll be discussing a brief discussion about that, and then we'll be solving your questions, your queries, and also the assignment questions. Okay, so let's move forward. So translation, as I have already told you, that it is basically the last step in the central dogma of biology. What is translation is basically translate means what? Decoding anything. Translate it into a meaningful thing. So this is what we are doing here also. We are translating the mRNA into a protein. It is just telling providing the information in a simpler, understandable, usable form, and that is protein, which will further perform its function as required by the cell or wherever it is required. So translation is actually the formation of protein using mRNA as template, and the formation will happen because of the presence of ribosomes and tRNAs. So here all the three types of RNAs are involved: the rRNA, tRNA, and mRNA. rRNA is actually the RNA which is involved in the ribosomes, which will be present in the ribosome. These rRNAs, along with the different proteins, will make the ribosomes, and those ribosomes will actually help in translating this mRNA into protein. tRNAs are those RNAs which will bring the amino acid required to form the protein and also help in the formation of a peptide bond. And mRNA is that RNA which has the information, which has the codes, which will tell us in what sequence the acid should be added so that the desired protein is formed. So that is how all of these RNAs are involved in translation, and that is how they are important. Okay, so let's move forward. Translation, if we'll go by the process, so translation process, the mechanism of translation is basically involves what? Activation of amino acid, initiation of translation, elongation of protein, and then termination of protein must be terminated also at some point where the sequence is completed, where the protein is completed. Okay, so for that there must be some procedure, that how does our ribosomes, sorry, come to know that this is the end of the protein. Now no further amino acid addition is required here. So that is the process of termination. Structure of translational machinery. What is the structure of translation machinery? First of all, we'll discuss about the structure of the messenger RNA, mRNA. This is the structure that we discussed when we were studying about RNAs also, when we were studying about this transcription part. So this is just a brief recap. So mRNA has 5' and 5' untranslated region, ribosomal binding site, coding sequence, and 3' untranslated region. So in eukaryotes, there are additional structures such as 5' guanine cap and poly A tail. Okay, so at the 5' and above the 5' UTR region, this cap will be present, and a 3' poly A tail will be present below the 3' UTR region. So this is just to protect the mRNA from getting degraded, from getting attacked by different enzymes. So this is a very important post-translational modification that happens in a mRNA. So mRNA has three reading frames, out of which only one codes for desired protein. This coding sequence, as you can see, that region that is marked as coding sequence will actually be the sequence of your protein that needs to be translated. But this coding sequence also has three frames as in the starting point and the ending point. And the frame is actually the three codon that will be recognized by the tRNA, according to which it will add the amino acid. So this has three, every mRNA has three reading frames through which it will read and form the protein, but only one makes the desired protein. So if in the sequence of bases there is no stop codon to interrupt the translation, then that synthesizes the entire polypeptide chain, and it is called as the open reading frame. Okay, if they don't have a stop codon, then it will synthesize whole of the sequence, and that will be called as open reading frame. So what is a genetic code? Genetic code is one of the important, we can say, is an important code, or we can say it is an important sign to tell us which amino acid should be added, and in what sequence. So genetic, because in what form you will send the RNA will send the information that in that sequence the protein is formed. This is all because of the genetic code. The genetic code is basically formed of three nucleotides. Okay, three nucleotides form a single genetic code, and every amino acid has some assigned genetic codes to them. An amino acid can have more than one genetic code, but a single genetic code codes for a single amino acid. It won't code for more than one amino acid. So this is something you guys should understand, and you should keep in mind. This will we will also revise when we'll study about the features of a genetic code. So mRNA is the random sequence of nucleotides differentiated by bases attached to them, the nucleotide bases, which are uracil, adenine, cytosine, and guanine. Three nucleotides together code for specific amino acids and are called as the codons. Okay? Three nucleotide makes a single codon. Why three nucleotides is a part of the codon? The genetic codon is triplet code called as the codon. It is known that we have only four types of nucleotides. We all know that. We have studied it when we were studying the structure of DNA. So we have only four types of nucleotides that makes the whole genome. It is also known that each codon consists of three nucleotides, which means that there are 64 possible amino acids. Yes, 64 possible amino acids. Okay, so this is how we came to uh, Dr. Hargobind Khurana came to the conclusion that yes, this genetic code should be made up of three nucleotide. Then only he did a lot of experiments. After that, he came to the conclusion that a genetic code is made up of three nucleotides, and each codon codes for a single amino acid. Now if we go by this combination that three nucleotides will make a single codon, then the possibility is 64 amino acids, which is again more than what the amount of standard amino acids is known, which is 20. So yes, this shows that the genetic codons, there are more than one genetic codons that codes for a single amino acid. That is how it was justified that 64 codons will code for 20 amino acids. Uh, this this is actually the table which shows that which genetic code codes for which amino acid. Okay, and also one of the thing that you guys should know, that UAA, UAG, and UGA are three stop codons. They do not code for any amino acid. These are stop codons. Whenever an ribosome reaches any of these codons, then they understand that this is the time to stop the protein formation, stop the translation. So these three codons are recognized as stop codons when the translation is happening. Genetic code properties. The genetic code is a triplet code called as the codon. Each coding sequence has a start and a stop codon to initiate and terminate the translation respectively. Okay, as I've told you, a start codon is there and a stop codon is there. The stop codon is UAA, UAG, and UGA, and the start codon is AUG, methionine. Most of the cases, in all of the cases, the start codon is AUG, methionine only. Okay, so usually start codon is AUG, which code for methionine, and stop codons are UAA and UAG and UGA. In some cases, starting codons are GUG or UUG also. So, if you can see it from the table, koi aaya hai. Yes. Where were we? So yes, in some cases, the start codons can be GUG or UUG. Okay, but in most of the cases, it is methionine only. The code is unambiguous, which suggests that code is for only one amino acid. As I've already told you, that every amino acid can have more than one codons for them, which codes those amino acid, but each codon will code for a single amino acid only. It won't code for any other amino acid. There is no comma or gap in the code. This is the way that the codes are written. No comma, no gap. The code is degenerate. This means that one amino acid has more than one codon. For example, phenylalanine is specific to two codons, UUU and UUC. Only tryptophan and methionine are coded by a single codons. Only these two amino acids are those which are coded by a single amino acid. Otherwise, all of these amino acids are coded by uh, more than one amino acids. Okay. Further properties are the codon is non-overlapping. For example, a codon has whatever sequence then they have given, then codons will be AUG, CUG. That means it won't take an nucleotide from the previous codon. It will be separated out. It won't overlap with the previous codon. Okay. So, genetic code is universal also, which suggests that genetic code and its meaning is common for all of the life forms on the earth. If AUG is coding for methionine in humans, then it will code for methionine in an insect also. It won't code for anything else. So it is universal. The codons are universal. It will code for similar amino acid in all of the species, all of the organisms present on this Mother Earth. Okay. So which suggests the genetic code and its meaning is common for all life forms. However, there are some exceptions to this rule. For example, UGA is a stop codon, but it codes for tryptophan in mycoplasma, spiroplasma. Some exceptions are there because biology is a science of exceptions. So yes, some exceptions are there. Otherwise, it is a universal rule that all of the codons which are assigned to whichever amino acid will code for that amino acid only. And mitochondria of several species. Similarly, CUG codes for leucine in general, but in yeast mitochondria it codes for threonine. It can code for threonine also, but in yeast only. Okay. The structure of translational machinery, transfer RNA. So transfer RNA or tRNA is a clover leaf structure in 2 dimensions and an L-shaped structure in 3 dimensions. So tRNA is 73 to 94 ribonucleotides in length. This is the length of a tRNA. A tRNA molecule consists of 5' phosphate terminal, an acceptor arm that ends in the CCA terminal at 3' end. So this CCA terminal is necessary for a mature tRNA so that amino acid can bind there and can get transferred from tRNA to the polypeptide chain. Okay, so it is an important modification that happens on a tRNA. D loop, which often contains dihydrouridine, anticodon loop, and T arm, which has T psi C, which where psi is pseudouridine. Okay, CCA sequence is important as it is important for recognition of tRNA and is also site for attachment of amino acid, as I have told you already. Transfer RNA continues. Each tRNA is specific to amino acid that it carries and it in CCA arm. And there are 30 to 45 different types of tRNA in prokaryotes and 50 types in eukaryotes, which suggests that there is more than one tRNA for single amino acid. For example, for glycine, there are two tRNA which are represented as tRNA glycine 1 and tRNA glycine 2. Next is the structure of the ribosome. So ribosomes are ribonucleoprotein particles that contain rRNA and proteins. Because of the presence of RNA and proteins, it is called as the ribonucleoprotein. Each ribosome is made up of two subunits. In prokaryotes, mitochondria and chloroplast of prokaryotes, there is 70S ribosome, which is composed of 50S and 30S subunits. In E. coli, 30S subunit consist of what? अभी हम प्रोक्रियोट्स के उसकी बात कर रहे हैं, राइबोसोम्स की बात कर रहे हैं, विच आर 70S. इन यूक्रियोट्स इट इज 80S टाइप. एंड जो 70S, सॉरी, जो 70S राइबोसोम है दैट इज कंपोज्ड ऑफ 50S and 30S subunit. Two subunits are there, a larger subunit and a smaller subunit. The larger subunit is 50S subunit, the smaller subunit is 30S subunit. Together they make the complete workable ribosome. If any of the subunit is missing, ribosome won't be able to perform its function. In E. coli, 30S subunit consists of 16S rRNA and 21 R proteins, and 50S subunit contains 23S rRNA, 5S rRNA and 31 proteins. So these all of these molecules together make this ribosome. In eukaryotes, there are 80S ribosome which consists of 60S and 40S subunit. Uh, Further ribosomes, in as I was telling you about the 80S ribosomes of the eukaryotes, two subunits, the larger subunit is 60S, the smaller subunit is 40S. 60S subunit contains 28S rRNA, the small 5S rRNA and 5.8S rRNA, and approximately 50 protein. That makes the whole large subunit of the 80S ribosome. The 40S subunit consists of 18S rRNA and 33 R proteins. Okay, they together make the small subunit. Uh, each ribosome is made up of two subunits that we have seen. The 70S ribosome has three tRNA binding site, the P site, the A site and E site. So P site is the peptidyl tRNA binding site, as you can see in the middle. A site is the amino acyl tRNA binding site in the right corner, and E site is a deacetylated tRNA, also consists called the exit site. Okay. Now we are not going into the details of this translation process. We are directly not details, but yes, we can touch some part of this, the mechanism of translation, basically the difference between the initiation of prokaryotic and eukaryotic translation. So in translation process, main difference between eukaryotes and prokaryotes is in the initiation process itself. Some major differences between eukaryotes initiation and prokaryotic initiation are as follows. In eukaryotes, there is only one start codon for eukaryotes, that is AUG, and that codes for methionine. And not N-formyl methionine. So basically, N-formyl methionine is the start codon in case of the prokaryotes. In the prokaryotes, the start codon is always N-formyl methionine, while in eukaryotes, it is methionine only. Eukaryotic cells needs more initiation factors than prokaryotes because it is much more complex than prokaryotic translation. Eukaryotic cell requires 12 initiation factors. In eukaryotes, process of association of mRNA with smaller subunit is more complex than prokaryotes. 40s first subunit identifies the 5' methylated cap of mRNA, the poly A tail, and then there is a scanning process involved wherein initiation codon is recognized. Okay, initiation codon will be recognized, and this recognition is aided by ATP dependent helicases and the hydro and that hydrolyzes ATP. So it is an energy requiring process. This recognition of initiation codon is also aided by Kozak sequences, the 5' ACC AUGG to 3' similar to Shine Dalgarno sequence in prokaryotes. These are actually the recognition sequence. In prokaryotes it is Shine Dalgarno sequence. In eukaryotes it is Kozak sequence. Okay. Elongation is carried out by ribosome in three different stages: the decoding, basically the recognition of the codon, then the peptide bond formation, the tRNA which has brought the amino acid, so it will form the peptide bond between the previous amino acid and the next coming amino acid. Translocation, in this case tRNA of A site is transferred to P site to make a space for next amino acyl tRNA. Basically, tRNA is bringing the amino acid, so it should have a proper place to sit where it can sit and transfer the amino acid to the previous formed chain. So that is A site. And this shift is also coupled with the ribosome movement along with the mRNA. So it will pull, when it will move towards P, it will pull the mRNA along with itself for moving forward and passing the mRNA through the ribosomal subunits. Okay. Then the translocation part. So the three things are necessary for translocation: deacylated RNA removed from P site, peptidyl tRNA move from A site to P site, and ribosome should move on mRNA one codon down, so next codon can come to A site. So this movement is very important so that next codon can be recognized and the protein formation can continue. Okay. Okay, and for termination के लिए भी यहाँ पे they need a lot of factors. The release factor 1, release factor 2, that binds to the ribosome near to the A site. Polypeptide chains are released from ribosome by the peptidyl transfer complex. Peptidyl transferase complex transfers the carboxy terminal residue of polypeptide chain from tRNA of P site to water molecule. Now the release factor and GDP are released and it tRNA also freed. So this is how the translation continues. Now 70S ribosome is unstable due to the presence of initiation factor IF3 and IF1 and ribosome recycling factors. As a result, 70S ribosome disrupts into 30S and 50S subunits and prepared for initiation. So this is how the termination happens. Now this was all about the prokaryotic translation. Now we are coming to the eukaryotic translation. So in eukaryotic translation, the process of mRNA encoded protein synthesis is a universal process that occurs in both prokaryotes and eukaryotes. Members of both the groups use information present in mRNA, which comes from the DNA by transcription, to synthesize proteins with ribosomes as machinery. Proteins perform a variety of critical functions such as enzymes, structural proteins, or hormones, and therefore they are crucial biological components. Okay, so proteins perform a lot of function, as I've told you in the starting of the presentation. That is why they need to be formed very carefully and in the proper sequence. If a single amino acid is wrong in the protein, then that protein will not be used by the cell, because this single mutation can cause a lot of problem in the protein and its function. That is why that protein would be discarded, it would be degraded and then discarded. So protein biosynthesis has a key role in disease, as changes and errors in this process through underlying DNA mutation or protein misfolding are often the underlying causes of disease, as I've told you already, because that can cause disease that would be harmful for the cell. So cell will either degrade it if it can recognize it, and if it remains unrecognized and it reaches to its point of action or somewhere else, then it can cause serious diseases. So, the process by which the sequence of nucleotides in an mRNA molecule directs the incorporation of amino acid into a person is called as the translation. In prokaryotes, transcription and translation are coupled. If you remember, I've told you in the last lecture that it is a coupled process in which transcription will happen and hand in hand translation will also go on, but that is not the case in case of the eukaryotes. The machinery required for translating the language of messenger RNA into the language of protein is composed of four primary components: the mRNA, the enzymes, proteins, tRNAs, and ribosomes. So, all of these components are important for formation of the new protein. Messenger RNA is a single stranded RNA molecule that is complementary to one of the DNA strands of the gene that we have read in the last lecture. So it is similar to coding strand, but it is complementary to the template strand. Okay. So during protein synthesis, a ribosome moves along the mRNA, reads its base sequences, and uses the genetic code to translate each codon into the corresponding amino acid. Okay, so ribosome moves along the mRNA as I've told you, and when it moves from one codon to next, basically when one amino acid gets added, it will move in the direction in such a way that it passes one codon and a new codon is there in the site for the addition of further amino acid. So tRNA, it has a D arm, an anticodon loop, a T-CC arm, and an amino acid acceptor arm, which should have a CC at its 3' end. Otherwise, the amino acid won't be recognized. Okay? tRNAs, the transfer ribonucleic acid, is a type of RNA molecule that helps to decode an mRNA sequence into a protein and is made up of a single stranded polynucleotide chain. It functions at specific sites in the ribosome during translation, which is a process that synthesizes protein from a mRNA molecule. Proteins are built from smaller units called as the amino acids, which are specified by 3 nucleotide mRNA sequence, जिसको हम क्या कहते हैं, codons. Each codon represents a particular amino acid and each codon is recognized by a specific tRNA. They are adapters between codons and amino acids, and here we are talking about the codons. Each tRNA has its corresponding amino acid attached to its 3' and that end also should has CCA in it. tRNA is also named sRNA, soluble or supernatant RNA, and adapter RNA also. Okay, so this is this is just again some information about tRNA. So the structure of tRNA can be decomposed into its primary structure, secondary structure, the cloverleaf structure, and tertiary structure, the L-shaped structure. In addition to usual nitrogenous bases, tRNA contains a number of unusual bases also. These unusual bases are important as they protect tRNA molecules from dehydration by RNAs when they are floating freely in the cytoplasm. So basically these are for the protection. The protection is just because of the methylation which happens on adenine and it makes it inosine, which happens on uracil and it makes it pseudouracil, which happens on uridine and it makes it dihydroxyuridine. This is just to protect the amino acids from getting degraded. Other unusual bases found in tRNA include hypoxanthine, thymine, and methyl guanine. ओके, स्ट्रक्चर हम लोगों ने पढ़ लिया है, द एक्सेप्टर आर्म, द सीसीए टेल, द डी लूप, द एंटीकोडॉन लूप एंड द टी आर्म. वी आर not going into the details of the structure. Ribosomes. So eukaryotic ribosomes are larger and more complex than prokaryotic ribosomes, which are 70s type. Ribosomes exist normally as separate subunits that are composed of proteins and rRNA. The subunits come together to form a ribosome when they bind to an mRNA near its 5' end. On binding to an mRNA, the ribosome reads the nucleotide sequence from 5' to 3'. So, this is the thing that you should keep in mind when the replication was happening. It was also in this direction 5' to 3'. The transcription was also in this direction 5' to 3', and the translation also goes in this direction only 5' to 3' direction, synthesizing the corresponding proteins from amino acids in an N terminal to C terminal direction. Ribosomes are located in the cytoplasm, either freely floating or associated with the endoplasmic reticulum. They serve to synthesize proteins. The ribosomes are ribonucleoproteins to which multiple ribosome proteins are bound. The sequence and the structure of the ribosomal components are conserved in all kingdoms. Sorry. Underlying the common origin of translational apparatus, the ribosome provides the platform for proper assembly of mRNA, tRNA, and protein factor. It consists of small and large subunits. Okay. So tRNA binding sites are E site, P site, A site. So the first tRNA that comes to an assembled ribosome and mRNA is comes at P site, the peptidyl tRNA site. The second one comes at amino acyl tRNA site, and then the tRNA which has provided their amino acid, then their function is done, their job is done, so they leave it through at E site, which is called as the exit site. Okay, so this is the types of rRNA that are present in the different subunits of this ribosome. So this is for your information. You guys should read it. Ribosome site for protein synthesis. So three binding sites for tRNA. A site, P site, E site, जैसा कि हम लोग अभी पढ़ के आए हैं. All three sites are formed by rRNA molecule in the ribosome. During elongation, the incoming amino acyl tRNA binds to the A site. P site is where the tRNA linked to the growing polypeptide chain in the is bound. E site is the binding site for unloaded tRNA prior to its release from ribosomes. Three main steps of translation again: initiation, elongation, termination. So we are again not going into the detail. These are just some of the steps in eukaryotes that are involved in the uh, along with the many initiation factor, these are involved in the step by step initiation of the amino acids. So the ribosomal dissociation, formation of 43s pre-initiation complex, formation of 48s initiation complex, and formation of 80s initiation complex. ठीक है? In this way they will assemble and they will start the process of translation. Okay. So protein factors required for initiation of eukaryotic translation are this is the list of the protein factors that are required before starting for the translation. So you guys can read this information again. We are not going into the detail because these are some of the important factors. If you are preparing for some kind of a competition like NEET or something, then this is a very important part. You must remember these factors. They may ask these questions. Then now the protein synthesis has happened. Now the ribosome must be restored so that it can again help in the formation of this protein. So for that, what they do is after the release of the polypeptide and the release factors like ribosome, it is still bound to mRNA and is left with the two deacylated tRNAs. To participate in the new round of polypeptide synthesis, these mRNA and tRNA must be released and the ribosome must dissociate into small subunit and large subunit. So if we want to recycle the ribosome, then all of the things that got attached to the ribosome that needs to leave the ribosome, also this assembly, this larger and the smaller assembly needs to be separated, so that it can be reused again. So this is the way to recycle the ribosomes. Okay. So now there are these things which are called as the post translational modification, again a very important aspect when we are studying about proteins, because these post translational modifications decide where the protein will go, what will be its function, how will this protein protect itself. There are many things, and all of these things are actually dependent upon the post translational modification of the proteins, because when the protein is synthesized, it is a very immature protein, and when it gets mature, it needs to be modified post translationally. So there are many types of the post translational modification. First is the phosphorylation. So phosphorylation is an important post translational modification. It is prevalent from bacteria to higher eukaryotes, sorry, sustaining as mainly two types. First, it acts to functionally regulate the catalytic activity of the protein by defining a rigid and permanent 3D protein structure. Secondly, temporarily phosphorylated proteins serve as anchors for other proteins substrate in signal transduction pathway. As such, it acts as a key keeper in the regulation of many cellular processes like cell cycle, cell growth, apoptosis, and regulation of signal transduction pathway. So yes, phosphorylation is one of the most important post translational modification that happens in case of the proteins, because it can help in the functioning of the protein. Functioning in a way, it can either activate the protein or it can either deactivate the protein. Just a Yes. So as I was telling you, it can either activate the protein or it can deactivate the protein. When a protein is attached with a post-translational modification, or we can say it is phosphorylated. So if we'll go by the cell cycle pathway, it generally helps in the activation of the protein. If you guys know about the CDKs, CDKs we have studied when we were studying about the cell cycle also. So when we were studying about the CDKs, so the phosphorylation was somehow helping in the further processing in the cell cycle. So it was there to activate the protein. But in some cases, the phosphorylation can deactivate the protein also. So it goes both ways. Either it can activate the protein or it can deactivate the protein. ठीक है. Next post translation, no, we're still continuing with the phosphorylation. So mechanism of phosphorylation is, in eukaryotic cells, phosphorylation is known to occur only at the side chains of the three amino acids that are either serine, threonine, or tyrosine. Okay. The three side the side chain, the amino acids to which side chains this phosphorylation can occur is serine, threonine, and tyrosine. This is because these amino acids harbor a nucleophilic group, nucleus loving group, that is a negative charge containing group. The terminal phosphate uh, the terminal phosphate group, that is on the universal phosphoryl donor adenosine triphosphate, serves as the point of nucleophilic attack from the OH group, which results in the transfer of a phosphate group to the amino acid side chain. And the magnesium ions act as a catalyst by chelating the gamma and the beta phosphate groups, resulting in lowering of threshold of phosphoryl transfer to the nucleophilic groups. So basically this is the process, this is the whole process in which magnesium also helps, and this OH group attacks nucleophilically on the phosphate, terminal phosphate group of the uh, sorry, on of the adenosine triphosphate. Why adenosine triphosphate? Because it is a universal phosphoryl donor, and this phosphate group that got detached from the ATP will be used to phosphorylate any of these three amino acid as mentioned above, the serine, threonine, or tyrosine. Next is the conformational changes can affect the protein in two different ways. Phosphorylation causing conformational changes in the phosphorylated protein. These conformational changes stimulate the catalytic activity of the protein, as I was telling you about. Either it can activate a protein or it can deactivate a protein. So any protein can be activated or inactivated by the phosphorylation. Phosphorylated proteins employ the neighboring proteins which have structurally conserved domain that distinguish and bind to phosphomotifs. So these acts as a recognition signal also. These domains are specific for diverse amino acids. Protein phosphorylation is a reversible process, post-translational modification basically, which is carried out by the kinases. So kinases will help in the addition of the phosphate group, uh, which phosphorylates, and phosphatases will help in the removal of the phosphate group, which actually help in dephosphorylating of the substrate. So these two types of enzymes make possible the dynamic nature of the phosphorylated protein. So the balanced concentration of kinase and phosphatase is very important for the cell, and it is also important for the catalytic efficiency of a particular phosphorylation site. So, phosphorylation is a very important post-translational modification. Next is the glycosylation. So, glycosylation, as the name implies, it is the addition of glucose group या फिर sugar group. ठीक है? So, glycosylation is a function, is a dire function of the biosynthetic secretory pathway in the endoplasmic reticulum and Golgi apparatus. Approximately 50% proteins characteristically expressed in a cell go through this alteration, which involves the covalent addition of sugar moieties to the specific amino acids. Mostly soluble and membrane bound proteins expressed in the endoplasmic reticulum undergo glycosylation. So, this post translation modification occurs either in the endoplasmic reticulum or in the Golgi apparatus, including all secreted proteins, surface receptors, and ligands. Moreover, some proteins that are transferred from Golgi to cytoplasm are also glycosylated. Okay? So, glycosylation again, post translation modification happening in either endoplasmic reticulum or Golgi apparatus. Next is the ubiquitination. So ubiquitination, this is one another post-translation modification where ubiquitin is added to protein. Ubiquitin is the eukaryotic protein coded by 4 different genes in mammals. These are the names of the genes. So while 4 different genes, when we'll understand the process of ubiquitin, then we'll come to know that there are different types of ubiquitination that are happening in a cell. So, depending upon that and depending upon the process through which the ubiquitination occurs, there are 4 different genes in mammals that code for ubiquitin. Proteins is made up of 76 amino acid and has a molecular mass of about 8.5 kilodaltons. So if any protein is attached with a single ubiquitin unit also, it will increase its molecular weight from whatever it was plus 8.5 kilodaltons. So it is characterized by the presence of C-terminal tail and 7 lysine residues. In ubiquitination, basically carboxylic acid of the terminal glycine from the diglycine motifs in the activated ubiquitin forms an amide bond to the epsilon amine of the lysine in the modified protein. So how it happens? The characteristic feature of a ubiquitin is the C-terminal tail and 7 lysine residues. These are the characteristic feature of any ubiquitin protein. अब यूबिक्विटिनेशन में होता क्या है, द कार्बोक्सिलिक एसिड ऑफ द टर्मिनल ग्लाइसिन, द बेसिकली सी टर्मिनल ऑफ द टर्मिनल ग्लाइसिन from the diglycine motif in the activated ubiquitin forms an amide bond to the epsilon amine of the lysine in the modified protein. Okay, it marks the cellular protein for the process of degradation via proteasomal degradation, changes protein's location, prevent or promote protein-protein interaction. Okay, so it can cause these kind of the changes in the protein. Uh, Next is the activation of ubiquitin. The steps are it occurs in a two-step reaction process. At first, ubiquitin interacts with ATP and forms a ubiquitin adenylate intermediate. In the next step, ubiquitin is transferred to even active site containing cysteine residue. This causes formation of the thioester linkage between the C-terminal carboxyl group of the ubiquitin and the even cysteine self-hydryl group. So the bond forms is thioester bond. Next is the transfer of ubiquitin from even active site to E2 active site via transesterification reaction. In the last step of ubiquitination cascade, there is a formation of an isopeptide bond between a lysine of the target protein and the C-terminal glycine of the ubiquitin via activity of one of the hundreds of E3 ubiquitin protein ligases. So this is the function of a ligase. So ligases are also of many type depending upon their structure and also depending upon how they transfer ubiquitin either from E2 or from its own self. Okay. So one E1 molecule causes binding to several E2 which in turn bind to hundreds of E3 in hierarchical fashion. Next, post-translation modification is the methylation. This process refers to the addition of a methyl group to the nitrogen, N-methylation, or oxygen, O-methylation to amino acid side chain. N-methylation is irreversible, whereas O-methylation is potentially reversible. Methylation enhances hydrophobicity of amino acid and neutralizes the negative charge when attached to the carboxylic acid. Main methyl group contributor for such reactions is S-adenosyl-methionine. This reaction is mediated by the enzyme methyl transferases. Methylation process is involved in epigenetic regulation as histone methylation and demethylation. Okay, this is something related to the activation or the deactivation of a particular gene from the histones. So there this post-translation modification comes into action and very useful. Okay, so this was what you guys must have read in the last week's lecture. I hope you understood this part clearly. If you still have any queries, we can discuss it in here this in this lecture. And if you don't, we can move towards the assignment discussion. So I'm waiting for you guys if you guys have any query. Otherwise, we'll move towards the assignment discussion. Any queries, anyone? Should I move towards the assignment discussion part? Okay. Okay, guys. So as our practice goes always, I'll ask the question. You guys will write the answers in the chat box. No one will speak up the answers so that everyone can perform. So the first question is AUG is the usual start codon which codes for the which amino acid? It is methionine. Okay. So the answer is methionine. Okay. Next question is the code is following which suggests that each codon specifies only one amino acid. Unambiguous. Yes. Third, the acceptor arm of a tRNA molecule ends in the conserved sequence that dash at the 3' end. What is that sequence? Question number 3, everyone. Yes, question number 3. Hmm. So yes, the answer is CCA. CCA is the one which will recognize the amino acid and through which amino acid will get attached to it, and this will then help in the transfer of amino acid from itself to the polypeptide chain. The bond here forms between amino acid and this CCA codon is isopeptide bond. Okay. Next question. Question number 4. Ribosomes are ribonucleoprotein particles composed of what? You can't see my screen, Bhavna. Check your connection. Is anyone else is also facing the problem? If they are not able to see my screen, now it is visible. दिबाद्रिता अभी आपको दिख रही है स्क्रीन? बढ़िया. Now answer for question number 4. Ribosomes are ribonucleoproteins particles composed of what? Yes, so the answer to this question is rRNA and proteins. Okay. Next question. In translation, amino acyl tRNA synthase is an enzyme that activates the amino acid and attaches them following to their corresponding tRNA. बताइए, what kind of a bond is formed there? Okay, so the answer to question number 5 is covalently, because it is forming what kind of a bond? Covalent bond, and the name of the bond is, what is the name of the bond? कोवलेंट बॉन्ड बनाता है, ठीक है, पर नाम क्या है बॉन्ड का? दैट फॉर्म्स बिटवीन टू अमाइनो एसिड्स. बिल्कुल स्टार्टिंग वाली स्लाइड में आई टोल्ड यू गाइस. इट इज द पेप्टाइड बॉन्ड दैट फॉर्म्स. Okay, so it is peptide bond. Next question. The Shine Dalgarno sequence following is located 10 base pairs upstream of the initiation codon. So what is the Shine Dalgarno sequence? Yes. The answer to this question is A 5' to 3' AGG AGGU. Okay. Uh, Next question. Question number 7. Following is the process of mRNA encoded protein synthesis. Yes, it is translation. 8. These unusual bases are important as they protect tRNA molecule from dash when they are floating freely in the cytoplasm. Question number 8, everyone. Yes, it is degradation by RNAs. ठीक है. Next question. The ATS ribosome dissociates to form which subunits? What are the two subunits that form by the dissociation of ATS subunit of ribosome? बताइए. The answer to this question is C. Yes, 60S and 40S. Okay. So answer is C. 60S and 40S. Next question. For the appropriate association of 43S pre-initiation complex with mRNA, energy has to be supplied by what? What is the energy supplier here? Question number 10. It is ATP. Yes, answer is right. It is ATP. Amino acyl tRNA is placed in the A site and dash are recycled to bring another amino acyl tRNA. What are that? Factors basically they are asking about. Yes, it is elongation factor 1A. ठीक है? Next question. During post-translation modification, magnesium ions act as a catalyst by chelating the gamma and beta phosphate groups, resulting in dash for phosphoryl transfer to the nucleophilic group. बताइए. Question number 12. Yes, it is lowering the threshold. It is the lowering of threshold of energy so that the transfer can happen. Okay, question number 13. Dash linkage forms between the C-terminal carboxyl group of ubiquitin and E1 cysteine sulfhydryl group. अभी पढ़िए हमने. What is that? Yes, it is thioester bond. It is thioester bond. Ubiquitin is characterized by the presence of a dash and dash. ये भी हमने अभी पढ़ा है. Yes, the answer is again correct. It is C-terminal tail and 7 lysine residues. Next and last question. Conformational changes stimulate the catalytic activity of proteins. So any protein can be activated or inactivated by. We had a discussion over this. I gave you an example of cell cycle. Yes, it is phosphorylation. So this is the end of your questions of the assignment. If anyone had any doubt with the questions, they can ask me. If anyone didn't understood anything, they can ask me also. You guys can ask me if you have any confusion in the assignment questions or in the lectures. Yes, anyone? Any questions? किसी को कोई क्वेश्चंस नहीं पूछने हैं? सबको सब समझ आया अभी तक? आप लोग को सब याद है? So, how methylation increases hydrophobicity? Basically, methylation does not have any charge. ठीक है? It does not have positive charge. It does not have negative charge, and it is a group that increases the bulkiness on whatever it is it gets attached to. When it increases the bulkiness, then it leads towards the hydrophobicity. So that is how the addition of methylation increases the hydrophobicity. ठीक है? हाँ जी, और किसी का कोई क्वेश्चन है? Okay, Bhavna has asked me a question. What is a dead box ATPase? So, Bhavna, dead box ATPase, I don't know much about it. I can read about it and tell you in the next lecture. But something I know about it is a highly conserved enzyme that binds and remodel the RNA. ठीक है? It helps in the remodeling of the RNA, and it acts as a chaperone. Uh, it is involved in various processes. This is the only thing I know about dead box ATPases. But yes, if you want, I can tell you in the next lecture. I can read about it and tell you in detail. This is the only thing I know, that they are acts as a chaperones, and they are very conserved. They are generally used to remodel the RNA. और कोई questions हैं किसी के? If anyone wants to ask any other question. Okay, if you don't have any questions, you guys can leave the meeting. Thank you for joining. Good night. Bye.